Yeah, this is something that Guru and I actually developed together. When Guru was still at Dana-Farber before he moved down, took his talents south to Florida. But the idea, is that ADCs are a chemotherapy with a very unique delivery mechanism. If we look at informative vedotin, EV, atezolizumab, govitecan, and SG, they're unique targets, unique payloads, and subsequently very different toxicity profiles. And they're both used sequentially in urothelial. But we combine chemotherapy all the time. Why can't we combine ADCs together? And that was the idea for what we call the double antibody drug conjugate, or DAD trial, right? So this was a phase 1 trial, really just to say, can we do it, right? And so we looked to assess the maximum tolerated dose of the drugs by looking at DLTs during cycle 1. And we enrolled 24 patients, which 23 started therapy. Median age was 70, and patients were up to 88 years of age on this trial. So these are not 50-year-olds, right? Up to 88. And what we saw is actually the maximum tolerated dose of the two drugs was actually full dose SG 10 mg per kg with full dose EV 1.25 mg per kg given day 1/8 of a 21-day cycle. Now, that being said, there were 3 DLTs and 5 patients evaluable. And so when we look overall at the cumulative toxicities, we think that the dose we want to move forward is a little bit lower dose. So 8 mg per kg of SG with 1.25 mg per kg of EV, day 1/8 of a 21-day cycle. If we look at overall toxicities, there were no new signals. We had diarrhea, neutropenia, anemia, what we'd expect with EV and or SG. There was significant risk in the first 6 patients. 2 patients had neutropenic fever, and subsequently allowed GCSF with cycle 1 at investigator discretion. And the majority of patients did get GCSF, which is not surprising. When we think about SG, that most patients would meet criteria for Neulasta with cycle 1. So that's great. It can be given safely. Does it work? And so these were heavily treated patients. Only 1 patient progressed on pembro alone. 40% had progressed on EV and on platinum and immunotherapy, and 40% had progressed on 3 lines of therapy. And what we saw was an objective response rate of 70% with 3 CRs out of 23 patients. Only 3 progressed disease as best response. And of the 23 patients, 9 had responses that are ongoing with 15 months follow-up. So really early data, but for me, it's super, super exciting. This is the first trial to show that ADCs can be given together in any malignancy, not just bladder cancer. So it has implications beyond urothelial carcinoma. Over a dozen ADCs are approved in urothelial carcinoma. The max tolerated dose was actually full dose. We don't think that should be studied, but we do think that the recommended phase 2 is a little bit lower dose of SG, 8 mg per kg, building upon the standard dose of EV 1.25 mg per kg, day 1/8 every 20-day cycle, is really interesting. So we have multiple expansion cohorts that are going to be opening at Boston, as well as with down in Florida and other sites across the country, looking at an expansion of the DAD, so EV plus pembro and short treatment resistant. And then what we just talked about, that really exciting data for EV pembro, looking to add SG to EV pembro, so the so-called DAD-IO or DADIO trial. So I think it's really exciting in really trying to sort of further advance care for our patients.
I think that Brad explained that quite well. So essentially, the rationale was quite simple. As you know, enfortumab vedotin and sacituzumab have different surface targets, Nectin-4 and TROP2, and also have different payloads. The MMAE, the auristatin, is a tubulin antagonist, and the SN38 in sacituzumab is a topoisomerase 1 inhibitor. So really, they have different non-overlapping toxicities, different payloads, different targets, surface targets. So it really made a lot of sense to us to combine these 2 agents, because they were both also actually approved and have single agent activity in urothelial carcinoma. So really the data look impressive, with 23 available patients. So obviously the next steps of further development and the addition of pembrolizumab would be very exciting, and potentially and hopefully will give us a further increment on EV pembro, which would be exciting if that happens. So it's really it's a novel direction and a novel paradigm to combine 2 ADCs. I wanted to highlight a large retrospective analysis where I was involved in looking at the correlation of safety and efficacy of atezolizumab, that's a PD-L1 inhibitor, across different solid tumors. This is a genetic database trials done by Genentech. So essentially, we looked at 14 clinical trials that assess atezolizumab, either alone or in combination with chemo. So chemo, IO, or IO. This is a big data set, 9,500 patients plus, 14 clinical trials, and a range of solid tumors in non-small cell lung cancer. There was triple negative breast cancer, there was urothelial carcinoma, there was RCC, small cell lung cancer. So the bottom line is that we already know that an immune adverse event is a pharmacodynamic marker for T cell activation, and it does correlate with improved outcomes. But what we wanted to tease out was, does the grade of the immune adverse event matter? Does really severe immune event, is that bad? Is a lesser severity immune event, grade 1 and 2, would that be preferable? That was a hypothesis going in, because obviously if you have a very severe event, you have to stop the therapy completely. Well, if you have a minor event, you can continue and you can sometimes resume. So indeed, that is what we saw. So basically, when we looked at it, so grade 1 and 2 immune adverse events were associated with improved survival with a hazard ratio of 0.65, whereas more severe events, grade 3 or 4 events, were not associated with improved survival. In fact, there was a trend towards worse survival. The hazard ratio was 1.16 and the p-value is 0.06. So nearly significant p-value for worse survival if you have a grade 3 or 4 immune adverse event. So really this teases out the importance of the grade of the event, immune event, that leads to an association with survival in patients receiving at least atezolizumab in this case. So I think this is interesting, and I think further work needs to be done in overall predicting an immune adverse event. If we knew who was going to have a grade 3 or 4 event, should we not give the check PD-L1 or PD-1 inhibitor? So I think that the interplay between activity and toxicity has not been well teased out yet. So I think we need to do some more work on this.
I'm standing in front of a poster that I was able to work on with an actual guru as part of this as well. So we've had a lot of advances in urothelial carcinoma. We just heard about ED-Pembro and Nivo, but all those trials exclude patients with predominant variant histology, right? So those patients who have small cell or adeno or urachal, like they're underrepresented. We don't know what to do with them. They're we have patients come to the clinic all the time, say, hey, I have this, what do I do? And so we sort of looked at that and said, hey, this is an unmet need for our patients. Immunotherapy is tumor agnostic. And so, several years ago, we developed a rare G protocol. We looked at the role of Nivo-Ipi in an unmet need. And one of those was variant bladder. And so we actually enrolled 19 patients. We saw 39% objective response rate, which was really exciting. And, compared to what's how a Nivo-Ipi at high dose Ipi and traditional urothelial carcinoma. So we went back and we actually engaged with BMS and we did an expansion. And so now we actually have been able to enroll 50 patients and treated them with Nivo and Ipi. A majority of patients progressed on prior lines of therapy. We enrolled patients with adenocarcinoma, small cell, urachal. And what we saw was a 37% objective response rate, which is really quite remarkable. Most notable is like the small cell patients had a 50% objective response rate, which seems to be quite durable, and there were no new toxicity signals. So, I think small numbers, but 50 patients actually is a huge number for these various histologies. And we hope that this will be, as we do more data, genomics and PD-L1, that this data will really lead to a change in care for these patients.
I will highlight the THOR phase 3 trial, which compared erdafitinib versus pembrolizumab in patients with advanced urothelial. This was post-chemotherapy second line setting. So essentially, this is of course an FGFR3, FGFR2 activating mutations of fusions. And the interesting finding in this study was that the overall survivals were similar for pembrolizumab and erdafitinib. However, erdafitinib had a higher response rate, was maintained at around 45% that we've seen in previous studies. But pembrolizumab was also active in this population. Response rate preserved at 21%, which has been the response rate seen with pembro in an intention to treat all-comer population. So at the end of the day, the relevance of this may not be so much in the current day and age, because we're not usually thinking of pembro in the salvage setting. given that Pembro has moved on to the first-line setting in combination with EV, and of course, Nivo has moved on with Gemcis. Nivo in the first-line cis-eligible setting in Checkmate 901. So really, maybe you will not face this situation too much where you have to select between Erda and pembrolizumab as salvage. But given, but if you are faced with that situation, I think that they both seem reasonable options to me based on the data, similar survival. But I think in the more symptomatic patient, maybe you want to pick Erda because of the higher response rate. But maybe in the less symptomatic patient, pembrolizumab is certainly a reasonable option given the fact that the survival seems similar in this trial. And there might be toxicities, different toxicities with these agents. So clearly you have to have a discussion with the patient before making a choice. But at the end of the day, I think that they're both reasonable options, one after the other in these patients.